After inoculation of the explant in the culture media, the culture tubes are put inside the incubation chamber. Inside the incubation chamber, we can control the temperature, light, and humidity. After a specific incubation period, the plantlets arises in the culture tubes. The plantlets are then transferred to the soil and which ultimately grow into new individual. Now, the measures to be taken for maintaining the species during the tissue culture. So this includes sterilization of the culture vessels using detergents, autoclave, then sterilization of the instrument like forceps, needle by flame sterilization, sterilization of culture medium using filter sterilization or autoclaving method, surface sterilization of explant using surface disinfectant like silver nitrate 1%, hydrogen peroxide 10 to 12%, bromine water 1 to 2%, sodium hypochlorite solution 0.3 to 0.6%. The whole procedure of plant tissue culture is to be carried out essentially under aseptic condition. So, the overall design of the laboratory must focus on the maintenance of asepsis condition. Secondly, the worker is also required to have proper knowledge of operating various equipment like pH meter, balance, laminar airflow chamber, etc. While performing the tissue culture experiment, there must present the first aid kit and fire extinguisher in the laboratory to avoid any mishap or accident. In addition, proper attention should be given while handling the toxic chemical, and all the chemical should be kept in correct labeled container and bottle. Now next is the basic steps for in vitro culturing of plants. This includes selection and sterilization of the explant. Suitable explant is selected and is then excised from the donor plant. Explant is then sterilized using disinfectant. Next one is preparation and sterilization of culture medium. A suitable culture medium is prepared with special attention towards the objective of culture and types of explant to be cultured. Prepared culture medium is transferred into sterilized vessel and then sterilized in autoclave. Next step is inoculation. Sterilized explant is inoculated or transferred on the culture medium using aseptic condition. Next step is incubation. The cultures are then incubated in culture room where appropriate condition, light, temperature and humidity are provided for successful culturing. Next one is subculturing. Cultured cells are transferred to a fresh nutrient medium to obtain the plantlet. And the last step is transfer of plantlet. After the hardening process, the plantlets are transferred to the greenhouse or to the pot. Now the question is, why this plant tissue culture technique is important? It is important because tissue culture is a very fast technique. Thousands of plantlets can be produced in a short period of time. Through this tissue culture technique, we can produce disease-free plants. Tissue culture can grow plants round the year, irrespective of weather and season. Production of new varieties is also possible through this tissue culture technique. Production of haploid can be produced through this technique. Some plants which do not multiply by seed can be propagated through this technique. Production of new improved varieties of crop can be produced within a short period of time by the application of plant tissue culture technique. By plant tissue culture, mutant can be induced in the culture and resistant mutant can be selected to produce resistant varieties.